Hello viewers, in my previous interactions with you on the molecules of life, I have discussed about water, carbohydrates and lipids. Today we are starting with another interesting unit on nucleic acids. In this unit 4, we shall be covering the topics given in the syllabus, which are components of nucleic acids, that is, pyrimidines and purines, sugars and phosphate, nucleotides and nucleosides, that is, nomenclature of nucleotides and nucleosides, structure of polynucleotides, structure of DNA, especially Watson and Crick's double helix structure, and RNA. We shall also be discussing types of RNA. Then we will go on to discuss difference between DNA and RNA, biological role of DNA and RNA, replication, transcription and translation, genetic code, which involves transfer of genes from parents to progeny. As you are very well aware that nucleic acids are naturally occurring chemical compounds that serve as the primary information carrying molecules in cells and make up the genetic material, that is, genes and chromosomes. Nucleic acids are found in abundance in all living things, where they create, encode and then store information of every living cell of every living form on earth. Nucleic acids, DNA and RNA, carry genetic information which is read in cells to make the RNA and proteins by which living things function. The structure of DNA, double helix, allows this information to be copied and passed on to the next generation. They are the energy currency in metabolic transactions, the essential chemicals, links in the response of cells to hormones and other extracurricular stimuli, and the structural components of variety of enzyme cofactors and metabolic intermediates. Let me now discuss the history of nucleic acids. The journey of the discovery of nucleic acids begins from the 19th century. Perhaps the first observation of a substance which was later identified as nucleic acid was made by the German scientist Justus Liebig. While developing the manufacturing process for making beef extracts, Liebig in 1847 reported the presence of an acidic material in filtrate from beef muscle. Liebig named this substance as inosinic acid. This name is derived from the Greek word inos, which means fiber, and more specifically that of muscles. It was more than 20 years later that a young Swiss physician, Friedrich Mischer, in 1869, isolated a distinct substance from the pus-coated surgical bandages. He chose leukocytes, that is WBCs, as his source material from these pus cells and first investigated them for proteins. However, during these experiments, he noticed a substance with unexpected properties that did not match those of proteins or any other previously discovered molecule. This molecule was very rich in phosphorus. He further examined the properties and composition of this enigmatic substance and showed that it fundamentally differed from proteins. Mischer then isolated a yellow solution of a substance from these nuclei. By adding acetic acid or HCl in excess, he could obtain an insoluble flocculent precipitate. Mischer noted that he could dissolve the precipitate again by adding alkaline solutions. Due to its occurrence in the cells, nuclei, he termed the novel substance nuclein, a term still preserved in today's name, deoxyribonucleic acid, abbreviated as DNA. Mischer also discovered the presence of nuclein in the cells of other tissues. Without knowing anything about how nuclein functioned, Mischer nevertheless assumed that it played a central role in cells. Mischer also realized that the presence of nuclein in the nucleus created an important chemical difference that set the nucleus apart from the cytoplasm. He was so convinced of the importance of nuclein for the identity of nucleus that in 1871 he even suggested that nuclei should no longer be defined based on their morphological properties, and that the presence of nuclein in them should be the criteria for their classification. According to him, nuclein more closely correlates with the nucleus' physiological function. Ironically, neither Mischer nor his contemporaries could at that time fully grasp the significance of this discovery. This paper on nuclein by Mischer, Felix Hoppe-Seyler and P. Plauze was published in 1871. In fact, Mischer had unknowingly obtained the first crude purification of DNA, and the significance of this discovery remained unknown to him during his lifetime. A few years later, Richard Altman, in 1889, separated proteins from nuclein and renamed acidic material as nuclein soure, that is, nucleic acid. Another scientist in Hoppe-Seyler's laboratory, Elbridge, started working with his students using hydrolysis and other techniques to chemically analyze the nucleic acids, thus discovering their component compounds. Kossel discovered that nuclein was made up of 4 bases: adenine, guanine, thymine, cytosine, along with sugars. Later, uracil was also detected. However, for long after Mischer's death, nuclein still received comparatively little attention. The vast majority of scientists remained convinced that more complex proteins must be the carriers of genetic information. Why were proteins preferred? Because proteins are made up of 20 different amino acids, while DNA is made up of only 4 different nucleotides. It was believed that 4 nucleotides seemed to be too few to store the enormous amount of genetic information in different living organisms. It was only after the work of Sutton and Boveri which established that nucleic acids were localized in chromosomes. Their chemistry attracted attention once again. A Russian-born American biochemist, Phobos Levine, has been another important investigator in this field. He studied the structure and function of nucleic acids and characterized the different forms of nucleic acids. Levine worked with A. Kossal and Emil Fischer and later identified the components of nucleic acids. Levine discovered DNA from thymus cell, and after Kossal's discovery, it was renamed as thymine. Levine and Jacobs in 1909 recognized D-ribose as a natural product and an essential component of nucleic acids. They also recognized that the unnatural sugar that Emil Fischer and Oscar Pilotti had reported in 1891 was in fact the enantiomer of D-ribose. Levine went on to discover deoxyribose sugar in 1929. His discovery led to the change in name from thymous nucleic acid to desoxyribose nucleic acid. Levine distinguished DNA from RNA and found that DNA contained adenine, guanine, thymine, cytosine, deoxyribose sugar, and a phosphate group. RNA was composed of ribose sugar, adenine, guanine, cytosine, and uracil. That means DNA and RNA differed in the sugars and the pyrimidine bases. That is, in DNA it was thymine, whereas in RNA it was uracil. Not only did Levine identify the components of DNA, he also showed that these components were linked together in the order phosphate, sugar, base to form units. He called each of these units a nucleotide. On your screens, you can see how a phosphate group is attached to a sugar molecule, which is a deoxyribose sugar, which in turn is attached to a nitrogen containing base. So this is how Levine showed that phosphate is linked at the 6th carbon, phosphate group is linked at the 6th carbon, and the adenine nitrogen containing base is attached to the 1st carbon of deoxyribose sugar. Levine is also known for his tetranucleotide hypothesis, which he gave in 1929. He proposed that DNA was made up of equal amounts of adenine, guanine, cytosine, and thymine. Levine stated that the DNA molecule, and I quote, that the DNA molecule consisted of a string of nucleotide units linked together through phosphate groups, which are the backbone of the molecule. According to him, if on your screens you can now see the tetranucleotide model of Levine, and he said that each unit of nucleotide is made up of a base which is attached to a sugar, which in turn is attached to a phosphate group. And according to him, was very interesting to note that according to him, this tetranucleotide model, the adjacent sugar molecules were connected by a 3', 5' phosphodiester linkage, and bases were attached to the C1' of the sugar. So as you can see on your screens now, according to Levine, the phosphate group was attached to the C5 of the deoxyribose sugar. And you know that when we do the numbering of the sugars, we number it one from here. To the 1' group, to the 1' carbon, the base, the nitrogen containing base, in this case we've showed adenine, was attached at C1', and at C6' we have a phosphate group which is attached here. It is forming a ester linkage. And when we look into the modern structure, the Watson-Crick model, what we find that another ester group is found here between the C3 carbon of the deoxyribose sugar. So this was the basic structure which Levine has proposed for nucleotide. The arrangement of bases, sugars, and phosphate in the nucleotide is identical to the most accepted model as proposed by Watson and Crick almost 24 years later in 1953. However, each 4 nucleotide components is a separate molecule and the bases are directed to the outside. This is how it differed from the Watson and Crick model. That is, in this case, the bases are pointing outwards and each nucleotide is considered to be a separate molecule. So now the thing was, when they found this, and it was later on found by 3 scientists, Rudolf Singer, T. Kapuson and E. Hammerstein in 1938, who showed that the DNA of many species had a molecular weight of 500,000 to 1 million Daltons. Thus, according to them, Levine's tetranucleotide must be viewed as a polytetranucleotide. The simplicity of this structure implied that nucleic acids were too uniform to contribute to complex genetic variation. Even Levine himself declared that it could not store the genetic code because it was chemically far too simple. Attention thereafter once again focused on proteins as the probable hereditary substance. And for this lecture, I had referred to the books by Brucker, that is Concepts of Genetics, by Watson, The Double Helix, by Pierce, Genetics, A Conceptual Approach, by Klug and co-workers, Concepts of Genetics. Now in my next lecture, we shall be discussing the further interesting history of this interesting molecule, that is DNA. Thank you.